Kia ora, no papakura aho, ko Sarah Killick aho, kia ora. So I'm a senior plant pathogens advisor at Auckland Council, and one of the projects I've been really lucky to work on is the Kauri dieback detector dog mahi. So I'd like to introduce or revisit probably that mahi for you today. So the detector dog project, we sought to train 2 dogs, Pip and Mawhai, to detect Phytophthora agathidicida, so that's the causal agent of Kauri dieback disease. And they needed to be able to discern it from other Phytophthora species and also non-target items. So after the training, the next step was to evaluate the dog's detection performance, just so that we could tell how accurate their detection is. The benefits of this project was that we endeavoured to produce a training and validation method that we could use for other dogs within our team to detect Kauri dieback, but also other councils could potentially use it, and we may be able to use the same method for other diseases that might come in the future. Importantly, we needed to quantify their specificity and their sensitivity. So specificity is that they are specifically detecting PA, Phytophthora agathidicida, and their sensitivity, excuse me, just how accurate they are at making those detections. It was our hope that the dogs could, or it is our hope that the dogs could be deployed as a fast screening tool when those traditional soil testing methods aren't practical or possible. So a key example of that would be at ferry terminals. Soil testing, it takes weeks to come back, and obviously the ferry is long gone by then, with potentially some contaminated soil on it. So we thought that the detector dogs, if this method works well, could be deployed to seek out PA in such a setting and give us a really quick indication on where there might be an issue to enable us to clean them up before the vehicles or equipment or whatever is transported. There are, of course, limitations with that, even from this stage. The main ones are that it's only appropriate under certain conditions. So they are not going to be replacing the soil testing in forests, for example, because there are likely there's some risk of false negatives or false positives. And part of our quantification process is measuring what their likelihood is. So it's a multi-stage, a reasonably big project, as I'm sure you can imagine. The first stages, the stages we've already started, include the dog training that's been completed already, and the first stage of testing. So that's a double-blind study in a controlled area to test their detection ability. We've just completed that last Friday, and then the data analysis and reporting from that. So in a nutshell, or in a glass jar, what we do is stick a series of oats in this picture here that have been inoculated or have not been inoculated, and some other stuff like some tasty kibble, gloves, and water. And we put them in these PVC tubes around a room. So the dogs have been trained to go around and sniff out the tubes, and then if they think that it's got PA, they sit like is illustrated here. So it's a double-blind study. That means that obviously the dog doesn't know where the PA is, but also the dog handler doesn't know where the PA is. And the person who does know, who is Zach on our team from Auckland University, he's behind a curtain. So the handler also can't see Zach, and so the dog and the handler can't react to any accidental body language of his. The samples are placed randomly. That selection was done before the study even started. And there are 120 runs in total. Each run has 12 stations. So those stations each have a jar, and the jars could include at random PA, that's the causal agent of Kauri dieback, PC, Phytophthora cinnamomi, Phytophthora multivora, or some non-Phytophthora, so that's oats, water, gloves, or dog food. I've got a couple videos here. The first one is of the training process, and the second I took last Friday of the actual study in action. All right, so this is seeking out, sniffing through the tubes to see if, oh, and that's a positive mark. There we go. So the dog has marked, so the dog gets a treat to positively reinforce that reaction. There we go. Another, yep, pretty sure it's P8. That's the mark. Going, this is Brian. You can see from the dog's faces, they really quite enjoy this mahi as much as we do. Okay, so sniffing out. Oh, and a mark. So this wasn't an actual study, this is just in a training, obviously sharing with the media in public stage. But in our actual test, there's no one else in the room. So there we go, that's a positive mark, and that's correct. There's PA in that tube. So going back, yeah, definitely thinks they can smell PA, trying to get a third treat, and is brought along to the next tube. We don't need to see that again. So the study, the data collection is complete, but the next stage is a statistical analysis. So I don't have that to share with you today. I can't say how well the dogs have each done. Then we'll be reporting the results, which will include the dog's ability to detect the PA in the oats. But the next step after that would be probably to test their ability to detect PA in soils. So that would be at more field realistic values or densities, rather than in the inoculated oats, which are probably quite highly concentrated PA levels. And then we'd hope to deploy the dogs potentially at some high risk locations like ferry terminals or potentially nurseries. This work is, of course, a shared effort. So my team, the Kauri dieback team, have been working on it with scientists Ellery and Zach from the University of Auckland. So Ellery's now with us at Council. Plant and Food Research, and Al Glenn from Manaki Whenua. And our dog trainers are Kieran and Brian. You might have seen them. They use quite a few different detector dogs. And of course, our fluffiest co-workers, Pip and Mawhai, who are just here. Okay, I will just stop sharing, I think, so I can see the chat, and I'm welcome any questions. Thanks.
Yeah, so just to read out one of the questions here. Why is the dog on a lead in the trials?
In the trials, so that was part of the study design, to walk the dogs past each station for a set period of time, and it was almost entirely for statistical robustness. The trainers have indicated that that's not normally how the dogs run, so that's something new to them. So whether we'd need to change that in the future or not, we'll have to see from the stats. But that just enables the dog and the trainer to go together to spend the same amount of time at each station, so that we can say, yeah, that's a read or a not read. The dogs have had a chance at each station equally.
There are no other questions, but unless anyone wants to pop one in now, would you like to tell us why one of the dogs was wearing shoes?
Yeah, I The shoes are cute, right? The shoes would be useful for hygiene purposes, but they've actually not been carried out through the actual study. So the dogs have been just barefoot for the trial themselves. The shoes would be important though for areas where we haven't trained them for forests or nurseries, but if they did go in nurseries, I think they'd wear shoes so that they don't transfer PA should it be there around.
Another question is, could the dogs be used to test PA in Ngāhere?
Right, so that has been a question that's quite a popular question. The trainer's viewpoint, and I agree with this, is that it's probably not where their strength really is. So in the Ngāhere, it's obviously going to be quite a lot more complex for the dogs. But we also already have tools for the Ngāhere. We have soil testing, and we know the accuracy of that. So the dog's strength really is in situations where we need to do more rapid testing, such as the ferries or transport sector at the border.
There's just another comment here from Sandra. Dog shoes are helpful on Rangitoto Island. Did you want to expand on that?
That would make sense, both from a hygiene perspective, but also I would imagine the scoriaceous earth on Rangitoto would be
pretty sharp.
A bit dangerous too. Is that what you meant, Sandra? I can't see the chat actually, sorry.
Yes, that's what she meant. Yeah.
Yeah, it is pretty rough for some soft paws.
Yeah, there's always injuries, aren't there? Okay, that seems to be all the questions.
Kia ora tātou. Can you see my presentation or?
Yes, all good.
Yep, okay, all right, cool. Tēnei te mihi ki a tātou I tēnei rā. Ka hui tātou ki raro I te karanga o tēnei hui, he whakawhiti kōrero. Ko hoki anga whakapau karakia te moana, ko rangi ka wareware, te maunga, ko ngā tuki matawhārua te waka, ko ngā pue te iwi, ko Rebekah Fuller tōku ingoa. Hoi anō, kia ora mai tātou. Today I'd like to provide an update, or at least give a brief overview of our Phytophthora agathidicida, a hot composting trial. And I would also like to take the time to acknowledge the steering group that included Ed Ashby, Tame Taratu, Patricia and Rory for providing valuable feedback. Oh, right. Cool. Just minimize that and then put this up. Okay. So as a bit of background to this research, there are a number of weed bins that are provided to the Waitākere Ranges landowners every March in the War on Weeds campaign, and this produces nearly 150 tonnes of waste. Because this green waste is from the Waitākere Ranges, it may contain PA or Phytophthora agathidicida, infected material. So the current procedure is to take this waste and the liner of the bin to landfill. And research in the lab shows that composting can provide the heat necessary to kill these PA propagules. So the benefits, the take, the reason for this research is to evaluate whether the heat generated by commercial composting reliably deactivates mature PA propagules. Okay. So there were some concerns about this particular project. The commercial composting facility is run by Living Earth. It's a huge place. It's on Te Motu Ahiaroa, which is commonly known as Pukatutu Island, and this is a site of cultural significance. So we work with mana whenua concerns as the long-term viability of having the waste come from one rohe and then to be taken to another rohe and to be treated on a culturally significant site. So we signed an MOU to allow the research to happen, and hopefully with the results, if they're positive, lead to a discussion of what the long-term treatment of the waste might be at another facility. And then we also talked about the containment because we didn't want Kauri dieback PA getting into other windrows and then being turned into compost and then scattered around in people's gardens everywhere. So in order to combat this, we looked at 2 stages of research. One is around the containment trial. So we can conduct this in the absence of PA to assess if the research setup would be appropriate. And we really wanted to know if the temperatures were high enough to kill PA in these commercial windrows. And the second part was to use inoculated Kauri roots to see if the commercial composting deactivates these propagules. And we looked at putting in a 12-week trial. So we'll test the viability of Kauri dieback or PA at 2 and at 12 weeks, those 2 intervals. So here we have the setup. These little bags here are 25, they're 25 micron fine nylon mesh filter bags, and these are heat sealed. So this will prevent the PA propagules from getting out. And for our containment trial, we just use green waste. And this is inside a larger bag, which is just there to protect these smaller bags because they are quite fine and quite delicate. So in terms of the containment trial, we dug a hole in the side of one of these windrows. These are, this is actually the end of the windrow, so it goes, into the distance there towards the maunga. And so what happens is we dig a hole and put the little bags inside, and with a rope tied them to this steel warata, and that's one of the parasites on the islands. So in terms of our results, we were looking at the sample bag integrity. So was our setup able to protect these little very fine mesh bags? So we had 4 different sample type bags, and this is what they look like after 6 weeks. They look a bit sad, but the seals were all intact. And we only had one where we had a problem where we had the sandwich of green waste in between, and there was a stick that poked its way through. So we decided not to have these sort of sandwiches of green waste on either side of the smaller bags. The temperatures got so hot that we had a temperature tagger inside one of these, and it pretty much disintegrated. So that kind of gave us an idea that it definitely was hot enough within these windrows. But we had to check this out properly. So what you see here is the temperature loggers logging the temperature, and we found that it was getting over 55 degrees, which is our standard. The propagules would probably die at 40 degrees, but we wanted to be sure, wanted to be conservative. So 55 degrees was our cutoff. And you can see for all the temperature bags that this happens within 12 hours, which is quite good. So we were able to say the sample bag integrity was confirmed, making sure that we didn't put that extra layer of green waste in. And the temperatures were definitely high enough to kill PA. So in terms of the trial setup, if we're going to put these, so each bag has 2 samples within it and a temperature logger. You see the little dark circle in there. And we had the windrow, 12 sample bags at a depth of 10 centimetres, and then 12 sample bags at a depth of 1 metre. And if you think about water going through our windrows, which happens often because they've got to water them to keep them at a certain moisture, and also there'll be rain. What we wanted to do was collect the water coming off that windrow. And so we created a bund with using concrete blocks, and then also using tarps, 1-piece tarps, and had a sump and collection tank in that water that comes off from rain and from watering these sites will be taken and put into trade waste. So this was last week. So this is our inoculated Kauri root trial. So here's our bun, there's the concrete there, those 2 big tarpaulins put over the top. And then this is fresh material being put down. And this is an image of, so the blue ropes are where we dug up to 1 metre and we measured that. And then the white ropes are just for the 10 centimetre ones. The windrow is turned regularly in accordance to what usually happens in this sort of commercial size area. And so we're going to have 3 turnings in the first 2 weeks, and then the row will be turned 5 times over the remaining 10 weeks. And we also have hygiene procedures as well. So a dedicated excavator will be used to turn the rows, and the hitch and boom will be brushed off and is disinfected, while the excavator bucket will be stored within the bunded area. And the samples will be sent to PFR, and which they'll be plated. Kauri root fragments will be plated on a selective agar, and yeah, and there's also a set of samples that will be kept in our biosecurity lab, but then also some down in PFR, so we can do a comparison to see whether it's actually killing the PA. Is there any questions?
I can read it out. There's a question for Rebekah. Is it likely that the site in Pukitutu Island will be used in the future? And what about the surface of the windrows? Will that be hot enough?
Yeah, so it's probably unlikely that the Pukitutu Island will be used, just because of that, the transport of those weed species coming across to another rohe, especially since it's a culturally significant site. I don't think that would be supported in the long term by mana whenua. So we'd be looking at, if it does work, maybe setting up a site closer to Waitākere, or hopefully within the rohe there. And in terms of the surface, we're going to, so the next stage after this, we'll be looking at the surfaces of all the windrows and just putting some temperature gauges, data loggers out there, just to see, sort of what happens over the time, probably March, April, which is when when we actually collect from the weed bins.
So are the normal composting facilities not hot enough?
Yeah, they are. It's just that if you think about it, it's quite a big windrow. They're sort of 2 meters tall. So it's just about whether, while they're doing their work, that all of the material is going to be subjected to that hot temperature. So that's something that we've got to consider, that maybe there's a special way that these the windrows need to be turned, that you take out the tops of it, put it on a certain place, turn it, and then pull it out, put those parts that were on the top into the middle, and then put the rest of the windrow back on. So those are things that we don't know, because we want to make sure that every single part of that windrow, which is huge, is in the center and is getting to that heat, the correct heat.
Thank you very much. I think that's the last question. Now,
Awesome. All right. Tēnā koutou katoa, ko Yue Chin toku ingoa, he Kauri dieback advisor toku tūranga mahi. So I'm Yue Chin, and I work at Auckland Council as a Kauri dieback advisor. And today I will be giving you a really brief update on our Kauri dieback survey in the Waitākere Ranges, and taking you through the epidemiological approach that we're taking for this survey. I'll give you a brief update also about where we are in the work. In the interest of time, I think there might be several slides that I might skip through very briefly. So let's crack on. Okay, so when we talk about Kauri dieback, they can really refer to several things. So to be very precise, I'm going to be really differentiating between Phytophthora agathidicida and Kauri dieback disease. So agathidicida, or Phytophthora agathidicida, is actually the pathogen itself. That's the causal agent of Kauri dieback disease. And relating it to the disease we know very well by now, coronavirus disease, or COVID-19. So that's the infectious disease that's caused by a virus, the SARS-CoV-2 virus. is that differentiation, and the disease itself is the abnormal condition associated with very specific signs and symptoms. So the pathogen is transported by soil and water movement. Anything that transports soil and water will potentially transport the pathogen, which is why we use a lot of, we invoke a lot of measures to help with Kauri hygiene. And it destroys Kauri feeding roots, reducing the nutrient uptake to the canopy of the Kauri and effectively starving the tree and causing dieback with symptoms relatively akin to drought stress. It's a really severe disease. So these symptoms, though, they can be quite cryptic and variable. So some trees may show very few bleeds. Again, like with COVID, you've got different people with different variety of symptoms that are very similar to what might be exhibited for other diseases like the flu, for example, fever and cough. But there are there's some specific symptoms that are relatively specific to COVID. For example, the loss of smell and taste. So the top two symptoms we use to differentiate a case of Kauri dieback disease in a Kauri are canopy dieback and basal lesions, like what you see here. This really sort of gushing bleed here. So we've run quite a few different approaches to Kauri dieback disease or Kauri dieback surveillance, and the methodology so far has been an incursion approach. So trying to find out where the pathogen or the unwanted organism is, and that's allowed us to really hone in and target areas where disease might be via helicopters or similar. And then we go in with field surveys to target those trees and take soil samples and find out the extent of disease symptoms in Kauri stands. So that's a delimiting surveillance approach. So then now in the Waitākere Ranges, we've actually gathered quite a lot of information about where that those soil samples have shown that the pathogen is present. So now we're really moving to a more epidemiological approach because we're concerned about protecting Kauri or the Kauri population as a whole. So we really need to understand the health of the population as a whole. So in terms of priority sites, we're moving to a more long-term monitoring approach where we are focusing on Kauri population health. However, in healthy sites, we will be still undertaking surveillance because we want to know if the pathogen has made its way into that area as well. So in, again, the Waitākere Ranges, we're looking at a randomized survey that's a sample, a sort of a cross-section of the Kauri population, and we're using remote sensing imagery that we just didn't have in the past. So moving on to how we've worked in the Waitākere Ranges, what we've done is that we've estimated a Kauri population with remote sensing. So we've made a map of Kauri across the ranges. And working with Manaki Whenua Land Care Research, we've used a suite of 60 different factors, for example, canopy structure, and we determine the location of possible Kauri points. So in effect, having a bit of a census of the population. So we know that the accuracy of this map was approximately 90% when we did this analysis. And it's really important to note that not all of these Kauri points are actually Kauri because it's a bit of a first cut. We're just trying to see what the technology can do. In fact, some of these we know that are pines because in our reference plots they weren't actually pine trees to help differentiate between pine and Kauri. So there's that, but the technology is constantly being refined, and over the years we will try to validate what we've got on the ground versus what we can analyze through the algorithms and improve that analysis. So also some Kauri had higher chances of being classified as a Kauri than others. So if you're a bigger tree with a more defined structure, you're more likely to be entered into the population census, I suppose, the map. And it will not take into account all trees. So things like Kauri, which are saplings and under the canopy, won't be taken into account. So it gives us a really good starting point anyway. And an independent way, which is really important, an independent way of estimating the population of Kauri, regardless of the health status. So the plan is to refine this Kauri mapping over time as we go back into the Waitākere Ranges and resurvey. So when we compare numbers across time, we can make sure that we are comparing the equivalent population estimate. So it's an apples to apples comparison. And also the power of remote sensing is that, as we collect more imagery over the years, we can lay them on top of each other like snapshots in time and detect change over time. So that's really powerful for landscape scale monitoring. And it's also useful for looking at other species as well, because we've already acquired the imagery. So then we randomly selected 2,500 points to be surveyed, and a subset of that to be sampled with soil samples. So those were analyzed at our lab with the soil bioassay, and also another subset of that was distributed to our collaborators, Narako Taketake, for further research into the soil, biochemistry, and other factors as well. So we finished the survey with a huge proportion or huge number of Kauri being surveyed and soil samples being taken. So then we note as well that not all the trees that we looked at in the field that we thought were Kauri points were Kauri. So that's to be expected, and that's again to help us refine our Kauri population map. But the aim of the game was to actually get a good representation of our Kauri population and understand the health of the population. So at each site and each tree, we were taking or surveying them for information around their health, like canopy health scores and basal bleeds, for example. And so we can understand, when we go back into the field the next time, we will be able to get enough information and compare them across time. So we're following them across their life. And then as we gather enough information, we've got, say, over 2,000 Kauri having been surveyed, we'll have enough power in our data analyses to detect small effects. So this single survey actually went into quite a lot of detail. We had 3 major directions of inquiry. So the baseline disease prevalence study, so the number of cases of Kauri dieback disease we wanted to figure out. So that, as we go back over time, we'll find out the next layer of prevalence and the next layer again. And so then that will help us calculate how disease prevalence or the number of cases of Kauri dieback disease changes over time. And so we can target interventions accordingly. The next study is focused around risk factors. So that helps us understand what is particularly risky to Kauri, because we've undertaken a randomized survey of both diseased and healthy trees. We can then start to unpick the health of Kauri and how that relates to the environmental and other factors as well. And the last study is the test performance evaluation study, where we're looking at the diagnostic test. So the visual assessment of Kauri, like these surveyors are doing, they're trying to figure out whether they're assessing the Kauri and whether it is a possible case of Kauri dieback disease or not. So that is the visual assessment, and also the soil bioassay, how sensitive and specific the soil bioassay is. And that's relatively analogous to what we're doing in COVID, where we've got the differences between the PCR test, which is quite it's the gold standard basically, versus the rat test, which is potentially much less sensitive. So that will help us improve our surveys over time. And so I'll probably have to skip through these because I'm running out of time. But I really want to thank the cast of thousands that had really contributed to the design, the review of the methodology. We spent a lot of time on the scientific rigor of the study. And so this entire process wouldn't be possible without these groups. So just briefly, the next steps. So at this stage, we've completed most of the data analysis. We still have the multivariable analysis of the risk factors to go. And that involves really complicated things like logistic regression models and things like that. But we're mostly done with most of the analyses, and also we're going to be drafting a report that's also mostly done. But now we're just working through processes of external co-author review to make sure everything is written as they were done. And another, trying to add another layer of stringency, we're going to go to external international expert review again and publish the report at the end through the Research and Evaluation Unit, or REMU, at Auckland Council. And that will be around late May, early June. And then accompanying that will be another webinar that we will share the link to when we've got the dates finalized. So that's really it. Any questions? Probably just dumbfounded silence. Yeah, there's quite a lot of detail in there, but for more information, you can go back to the webinar we did last year, and that will give you a lot more detail of what studies we actually did. And yeah, there will be a report coming out in the near future. Sam from Auckland Park says, is there any research into the methods of spread, such as impact of foot traffic? So Sam, this survey itself, it's basically an observational cross-sectional study, which means that it's a snapshot of the health of the trees in time. So what we can do is look at correlation, but not necessarily causation. It's just the design of the study will not allow for cause and effect analyses. But what it will help us do is pull out the risk factors that potentially are correlated with disease and the presence of the pathogen. So then we can investigate them more and start to delve into those cause and effect scenarios. Also, we have quite a lot of things in the ranges and just in the general forest ecosystem environment that potentially will confound each other. For example, ridges versus slope and other things like that. So we're having to unpick all those confounders and build a really good model that can help us understand those correlations. So in essence, the impact of foot traffic, we've definitely collected information about that. We're definitely putting some layers or GIS layers of on track and roads and things like that into the model, but it still remains to be seen whether they are definitely statistically significant or not with this study. And then Kate mentioned, to what extent is it likely that the pathogen is present in soil and trees become susceptible due to trigger factors that are location specific? So it's pretty likely, Kate. The pathogen, it depends, depending, there's various interactions. There is a very clear host pathogen environment triangle where, if the environment is unhealthy, for example, or the host is unhealthy, then the pathogen might cause, more virulent symptoms of disease, I suppose. And also the environment could promote the pathogen to become more virulent and cause disease more easily. So there's various interactions going on. So trying to unpick that, and it depends obviously on the location as well. Certain locations, for example, you might be on the coast and you might have or a higher elevation, you might have poorer soil, so that might cause you to be less healthy. So it's just deconstructing those variables and trying to unpick what the trigger factors and, like you say, or the risk factors are. I think that's probably it from me. Nigel, do you want to wrap things up?
Yes, thank you, Yue Chin, and thank you to the Plant Pathogens team that have done such a great job today.